Alright, so in this lecture, I'm going to be talking about biomedical event extraction based on knowledge-driven tree LSTM. So I'm going to be explaining this paper in 2019. Event extraction for a biomedical domain is more challenging than in the general news domain or even finance, for example, since it requires broader acquisition of domain-specific knowledge and deeper understanding of complex contexts. To better encode contextual information and external background knowledge, we propose a novel knowledge-base-driven tree structure long, short-term memory networks, incorporating the two new types of features: dependency structures to capture wide context, and entity properties, which are types and category descriptions from external ontologies. The same thing that we do for entity linking. We use these information to train it jointly, and they evaluate it on BioNLP shared task with GenAI dataset. So you see, you have gene ontology, and here we have a tree LSTM. But this is a new kind of tree LSTM. It's a knowledge-base driven tree LSTM. And these yellow circles denote the external knowledge base concept embeddings. For biomedical event extraction, we mainly explore gene ontology as our external knowledge base, since it provides detailed descriptions for each gene and gene product attributes across all species. It consists of two types of information. The gene ontology defines all the gene functions, relations between these genes, functions, and aspects used to describe the gene functions, including molecular function, cellular component, and biological process. The gene product annotations provide all entity-related attributes, such as the fully entity name, entity type, as well as the gene functions it is related to. For example, given the entity tags from the gene product annotations, we can get its full entity name as tax protein, which is a type of proteins, and it's related to a function about biological process. From the gene ontology, we can further determine the specific function that tax is related to positive regulation of transcription in terms of biological process aspect. So we assign a word embedding which pre-trained on PubMed and PMC texts to represent each entity type. For a gene ontology function, which is usually a long phrase, we use a state-of-the-art sentence embedding to automatically learn a vector representation. We then concatenate these two types of knowledge-based property representations as the final knowledge-based concept embeddings. So given a sentence, for example, we first perform the dependency parsing you can use a Stanford dependency parser, Stanford Core NLP, or even Allen Institute for AI, or even Spacey, or even SciSpacey. For each node in the tree structure C of J, this is the set of children of node J, and mu sub k is the knowledge base concept embedding of node K. So we set mu sub k to 0 if node K is not a biomedical entity. Mu sub j tilde denotes the sum of these concept embeddings. So we incorporate the knowledge base concept embeddings into input, forget, and output gates of 3 LSTM. Besides adding the knowledge-based concept embedding into 3 gates to select the knowledge-based information, we also introduce knowledge-specific output gate. This is knowledge-specific output gate to explicitly incorporate knowledge information into each node's hidden state. So we eventually combine the implicit way of incorporating knowledge-based information into input, output, and forget gates, and an explicit way of directly incorporating the knowledge-based information into a node's hidden state. And CJ here is the memory cell. These WC and W sub mu are weight matrices that we should be learned. After getting hidden states of each node, we use a softmax classifier to predict the label for each node and optimize the parameters by minimizing a negative log likelihood loss. After detecting all candidate triggers, we further extract argument for each trigger. So given a trigger, for example, transcription, and a candidate argument, for example, this is the argument, we first perform dependency parsing and extract the shortest dependency path between that argument, which was transcription, and our argument, that word that was trigger, which is transcription, and our argument, which is OBF1. Of course, for shortest path, you can use Dijkstra algorithm, which is a very old algorithm, and obtain the shortest dependency paths. We use the same knowledge-based driven 3 LSTM to encode each node into new hidden state representation. We use the hidden state of the root as the overall vector representation of the whole dependency path. Finally, we feed the concatenation of H0 with the hidden state of trigger and argument as input to another softmax to predict the argument row. So here you see that task 1 is about trigger labeling. After you get the trigger, you do task 2 is argument row labeling. Because you already know the trigger, so you just need to know the rows. And of course, we use knowledge-based driven 3 LSTM. And this is Genia event extraction. You have different event types. Some of them have these protein, but you see these 3, not only they have protein as an argument, they also have event as an argument. For example, if the core argument is cause, not only they accept protein, but they may also accept the event. So it's a nested event structure. Nested, so this is an event by itself, this is a trigger. And this is another trigger for a different event. So it's a nested event structure. And you see the hyperparameters, the learning rate is not too small. Batch size is 25. The hidden size is very small. And they use type embedding size of 200, same as word embedding size. But the sentence embedding size is 4,000. You see the F1 score is pretty low, so that's why for BioNLP 2011, still it's challenging. State of the art is less than 75%.